Hello, my name is Lupita Barrera, and today we will be exploring gel electrophoresis and Western blotting. These are analytical techniques that are used to detect specific proteins in a given sample of tissue. Electrophoresis determines the presence and/or relative abundance of a given protein, and Western blotting is a method used to quantify the amount of a protein. We are performing these procedures to determine if the protein RAGE, which stands for receptors for advanced glycation end products, is existent in avian and rat kidneys, the rat kidneys being the control. You see, avians are a particular species because they have naturally high glucose levels, but they don't experience any of the complications that humans do under those conditions. If humans have high glucose levels, the glucose binds to albumin in their blood and form AGEs, which could then cause swelling and other complications. Our lab hypothesizes that birds may not experience this because they lack AGEs. Today I will show you how to run a gel electrophoresis in order to determine if RAGE is present in avian tissues and samples. We will begin by preparing the gels. These are the reagent and materials used to prepare both the running and separating gel. After we place the gels into the chamber, we carefully place the prepared homogenized samples into the wells. Now, we will apply electronic current. This will cause the smaller proteins to progress farther down the gel than the bulkier proteins. We will now transfer the gel onto a PVDF membrane. We will add then blocking buffer, a primary antibody, and a secondary antibody. The secondary antibody emits a signal that can be detected by a special camera. Here in the darkroom, an image will be produced using a special film. The fluorescence given off by the primary and secondary antibodies can be detected on the film, thus allowing us to use the densities of the band as quantifiers. From the results of the Western blotting, it showed that there was a presence of the RAGE protein in the avian homogenized tissue. Bands were detected in the 10 UG lane, 20, 30, and 40 protein lanes. When the mourning dove and rat vasculature were compared in a Western blot, it confirmed the presence of RAGE in avian vasculature. Our results would be more concrete if we ran additional Western blots using various concentration, new equipment, and fresh tissue. Also, we plan to run more immunochemistry and immunofluorescence on avian and rat tissue to detect the presence of RAGE in different types of tissue. Also, investigation on whether other proteins or enzymes present in avian plasma affects the AGE to RAGE pathway. Thank you.